Okay, our next speaker is Eric Ahrens, who will tell you about some of our nascent attempts to learn how to follow cells once they've been transplanted. So, Eric. And your stuff is on the main computer.
Yeah, I'm going to use this. I got it right. So good morning. Thank you for coming this morning. I'm going to tell you some stories about how we've been trying to image cells in the body using MRI. And as you saw in the last talk, there were some beautiful MRIs that are done in conjunction with these treatments, and it very vividly shows the anatomy in fine detail and where the lesions are. We'd like to add another layer of information and get a richer data set from the MRI. What we're trying to do is adapt MRI to actually then see the cell graft and what happens to that graft, what the fate of that graft is. And that's important for scientific reasons. We'd like to be able to understand if the cells survive, what the effect of anti-rejection drugs are. Is that really necessary, for example? And then what happens to those cells during the recovery, hopefully the recovery procedures. And so I'm going to talk about some sort of, I would say, disruptive technologies that we've been developing over the last 10 years or so. I have a disclosure. And a lot of the motivation at a high level comes from looking at websites, for example, clinicaltrials.gov. And you can just search for stem cells, and you see that there's almost 2,000 trials pop up that are currently open. And so this is not a little niche, an esoteric endeavor. It's this sort of activity of tracking the cells in the body can be used by many investigators all over the world. And so we're developing tools to enable that. We believe that cell tracking in vivo can accelerate clinical trials by helping you to optimize the cell delivery. So we saw very nicely in the last example how they devise these special apparatus, biomedical devices for efficient delivery of the cells. But you would like to be able to see if how well they work, how good a job the surgeon does in the actual delivery. Do we need to maybe make improvements? So for example, if we think we deliver 1 million cells, do we actually deliver 1 million cells? Maybe it's 100,000 cells. So we'd like ways to validate it quantitatively, the delivery. The dosing. If we are inefficient in delivery, we may have to add increased numbers of cells to get the target amount to repair the lesion. And so we'd like tools to actually be able to measure the dose after the fact. The modes of action of the cell, again, scientifically, what are the cells doing? How are they behaving? This helps people design, maybe go back and do a few iterations in the design of the cells. And we think that regulatory bodies like the FDA would like to know where the cells go after they're delivered. And we think that this can help lower the regulatory hurdles to getting some of these treatments approved in the future. If you have a way to survey and keep track of the cells, this will improve your safety. The regulatory bodies will be happy, and it will accelerate your clinical trial. Now, if you're contemplating imaging cells in a patient, you don't have many options. If you're going to image cells in a mouse or a $20 rat, you have many options. You can sacrifice the rat, take a necropsy, run flow cytometry, do histology. But as soon as you move to a larger species like a monkey or if you want to work in humans, you have much fewer options. So this is just a summary of different imaging methods. And these 5 are used clinically: MRI, SPECT, PET, CT, and ultrasound. In terms of cell tracking, though, only the first 3 have been contemplated: MRI, SPECT, and PET. The only clinically approved agent for tracking cells in vivo in patients is a SPECT agent, indium-111, and there's a technetium probe as well. But these are radioactive probes. And these aren't ideally, my view is these are not ideally suited for regenerative medicine applications because to label the cells, you need a standalone GMP lab to prepare the cells. And these materials like technetium are highly toxic. And if you want the cells to survive for long periods of time, you may have some concerns. What they're approved for is diagnosing inflammation. You do a leukapheresis procedure, take the white cells, you label them, you put them back into the body to diagnose a site of unknown inflammation. That's sort of an acute diagnostic application. But if you want long-term survival of the cells, those materials aren't probably the best. There's also issues about specificity and leakage of those materials. So PET is another method that people have been contemplating, and it's a very sensitive method, and you can devise ligands that target particular cell populations in vivo. The trick is, though, the chemistry and the target specificity. If you inject the material systemically, some fraction has to find the cells that you implanted to light them up. The other option is to label them ex vivo in a GMP lab, but those have the same issues that we had with the SPECT agents. So what we've been looking at over the years is MRI. And we've been working from sort of the ground up to devise imaging probes or tags that can label the cells, rendering them distinct from the background tissue, and so that you can see your cells of high specificity. There's been a lot of prior work, or some prior work, using MRI to track cells. They render the cells distinct using off-label uses of clinically approved contrast agents, such as gadolinium and iron oxides and manganese. These are off-label uses, but they're not, we believe they're not optimal for cell tracking. When you're doing the imaging with these agents, you have an issue with specificity, and that you have to take an image before and after scanning, and after the injection of the cells, and you have to then analyze differential contrast. Also, it's difficult to do quantification of cell numbers. So we've been looking at a disruptive technology using fluorine-19 as an alternate nucleus for MRI, and we do labeling with materials so-called perfluorocarbons, shown here. And this is sort of sampling of perfluorocarbons. These molecules have been used for many years in the context of artificial blood substitutes, and the safety profile is very good. And so we start with molecules that look like these or that look like these, that have a large number of chemically equivalent fluorine atoms, and yielding a high degree of sensitivity. Now, the advantage of using these is that when you do your MRI, you tune it to the fluorine-19. There's no background signal. And that enables you to see the cells unambiguously. You can then take a proton scan, like what we just saw, to look at the anatomy, and you overlay the two data sets. And you get a high image of high specificity. Now, the way the workflow is that you take your cells of interest. They can be stem cells, they can be immune cells, for example. You then add emulsified perfluorocarbon into the culture media. They're engineered to be taken up by the cells. They're internalized. Viable cells only take it up. It takes about 12 hours or so to label the cells in culture. And then you inject them into the subject, and you take an MRI scan. You take a proton scan for the anatomy, a fluorine-19 MRI scan, and then you overlay the two data sets. And this gives you the labeled cells in the anatomical context. And then you can draw regions of interest around areas you want to count the cells, integrate up the signal, and then analyze your data in that fashion. So this is what a labeled cell looks like. This is a dendritic cell, immune cell, and you see little droplets of the perfluorocarbon inside of the cell. And they're often taken up and trafficked to low pH compartments like lysosomal vesicles. This was the early data showing that this works. Looking at cell migration, for example, you can inject cells. They migrate to the lymph node here in the popliteal region, popliteal lymph node. You can also look at biodistribution of cells. If you inject the cells IV, you have no knowledge about where they go a priori, but in a single MRI scan, you can then find where concentrations of these cells reside. Now, you can't see single cells. You see where populations accumulate. So in this sort of technology, you can see about 10,000 cells per voxel using these preclinical scans in these rodents. Clinically, the magnet is less strong, so you can see fewer cells per voxel, something of order 10 to the 5th per voxel. And we've done many cell types. That was dendritic cells. This is T cells, where we can, in this model of type 1 diabetes, we can inject the T cells systemically. We can then watch the homing of the T cells of the pancreas in this pre-diabetic mouse, and then we can integrate up the signal and quantify the number of cells in the particular region and determine the efficiency of trafficking of those cells. You can do stem cells. You can inject them into regions, for example, in this stroke brain, in this hemisegment, and you can see whether you hit the right spot after the fact, and then follow those graphs days and weeks later. Now, as I said, these materials have a track record of being put into man, and so we've been working very hard on clinical translation. And so I'm going to talk about a trial that was published last year, and then another trial that we're trying to launch this year here at UCSD. The first is, I'm going to talk about a clinical dendritic cell-based vaccine to treat colorectal cancers. Now, the idea here is that you take monocytes from the patient from a leukapheresis procedure, you incubate these cells in a cocktail of cytokines to create immature dendritic cells. It takes about 5 days. And at day 5, you add activation factors and you perform antigen loading of tumor lysate that you acquire from the surgery in the intervening days. And then you also add this perfluorocarbon in the tissue culture lab as another factor. And then you let all that co-incubate for 2 more days. And then you, at the end, you get mature dendritic cells that are activated, loaded with antigen, and labeled for MRI. You then can cryopreserve those cells, and you do a release criteria testing to make sure that there's sterility, that the cells have the correct phenotype by flow cytometry, and the cells are not apoptotic. So you can do all that while they're frozen. And this is an example of how we would characterize cells preclinically. We look at things like cytotoxicity using trypan blue or NAO. We look at cell phenotype, looking at the maturation markers, CD86 and CD83. We look at cytokine secretion. We do mixed lymphocyte reactions where we put the human DCs with human T cells, with chemokine receptor expression, transwell migration assays, also antigen processing and presentation. In all cases, labeled versus unlabeled. The first question the biologists have or the clinician has is, what's this do to their cell? So we have labeled versus unlabeled. We run the same panel of markers and assays. And in all cases, it's been looking really great. This is in vivo data in a xenograft model. This is the implantation sites of these cells in the quadricep muscles. Immature DCs on the left, mature DCs on the right. These are expected to migrate. These are human cells. Here, and at day 18, the same animal, you see indications of migration. That's best seen here. We look at just the fluorine channel. We strip away the proton image, just look at the fluorine, and this is the lymph node here. The cells have migrated. You can take it out surgically, run flow, and prove that the human cells have migrated. So we presented this work to the FDA around 2006. This is a concept at that stage. And they suggested we pursue a drug master file designation for this agent. And this is a neat regulatory mechanism. And I believe it's the first time that this mechanism has been used for a contrast agent for MRI. Because you're using such a small amount, it's essentially considered an excipient. The IND then is held by the institution running the study. They cross-reference the drug master file. In this particular trial, we did a phase 1 pilot clinical trial against colorectal cancer stage 4. We're using autologous dendritic cell vaccine. We published this last year. And then this is an example of the patient data, the in vitro assays. So while these cells are frozen, we run tests to make sure they meet a criteria. We measure the cell labeling in individual patients. And this is a compilation of the cell labeling. So we can actually measure the number of fluorine atoms taken up per cell. And you see there's an outlier here, for example. But the average here is about 5 times 10 to the 12 fluorine atoms per cell. We look at cell viability. And then we look at these markers via flow assays. And what we do is we save some part of the cell that's unlabeled to serve as a control from the same patient. We benchmark the sensitivity of the scanner, and this is important. So we're working a 3 Tesla clinical scanner, and we run phantoms where we put 10 million cells, for example, in the bottom of a tube, and we bed it in a tube of agarose. This is the hardware modification that we have to do to the scanner. We have to run fluorine-19, so it's a little bit of an electrical circuit you have to buy. And then we can benchmark the sensitivity. And the voxel signal-to-noise ratio is on the y-axis. The number of cells in each voxel is shown here on the other y-axis. A voxel is the smallest volume element of an image. It's a 3-dimensional pixel. And this dashed line here represents our estimate of the sensitivity of this technique, which is about 100,000 cells per voxel, which is a conservative estimate. Then here, this is what data looks like. This is an NMR spectra of cells that have been injected into a patient. These are 10 million dendritic cells injected in the quadriceps. And then this is typical patient data. You can see the injection site. So you can see if you hit the right spot. And then, moreover, you can look at the fate of those cells. So you can actually do cell quantification in vivo. And in these patients we were able to quantify, you can see that there's approximately 10 million cells at 2 hours post-injection. But by 24 hours, half those cells have gone. So this is sort of new sorts of readouts. You can actually quantitate what happens to those injected cells. Then the most likely reason of why they diminish is because the cells die. The other option is that they've migrated away to peripheral tissue, including lymph nodes, although we didn't see any cells in the lymph nodes that were captured in the field of view of these patients. So this is, again, a platform technology. We have a drug master file. We'd like to now apply it to many different types of clinical trials, and that's a lot of my work here. I'm starting interactions with clinicians to apply this to cell trials, including stem cell trials, but also immunotherapy trials such as T-cell trials. One thing we've been working on now, we've sort of going to launch this year, I'm very excited about, is looking at treating myocardial infarct and remodeling using stem cells. So, of course, we all know that myocardial infarct is a major disease killing, one of the largest killers in the US and elsewhere. It starts with acute infarction, a disruption of the blood supply, and this leads to fibrosis and remodeling and ultimately heart failure. We've been trying to get this trial off the ground. It's using an autologous cell mix. It's manufactured by Vericell. It's a mixed population of cells. It contains you start with bone marrow through an aspirate. You then expand it using a proprietary process to make this cell product. It contains monocytes and mesenchymal stem cells, MSCs. And so this is the cell product. And these are injected transcardially with a catheter. So they snake a catheter up the femoral artery and inject the myocardium directly. We're going to do longitudinal MRI to look at the fate of the injection site. The primary imaging endpoints will be graft persistence. So one of the big questions with these types of therapies is how long do the grafts persist? Do they stay there for weeks or for longer? There's some possibility that there's engraftment because of the potential differentiation fate of the MSCs to form muscle cells. And so, but there's also probably a paracrine effect where locally they'll secrete factors that tamp down the inflammation and promote regeneration, potentially from the endogenous cardiac stem cells. So these are questions that people would like to know the MOA, but there's no way to visualize the behavior of the cells after the fact. And so we think this technology would be ideal. We're injecting 10 million cells like what we did with the prior study, so I think we'll be successful. There's also been a prior multi-site phase 2 authorization here at UCSD, so that I think minimizes the risk of having having difficulties getting it authorized. And we're getting seed money from the Sanford Stem Cell Clinical Center. We're very grateful for that. Prior work by some colleagues of ours up in Robarts, up in Canada, have done human MSCs, 1.5 million cells. This is a mouse. And you can see that they can do longitudinal studies with these cells, quantify the amount that are there. And we can then, so we draw on this, so we're pretty confident this will work. We've been able to image the fluorine in the heart. This is in a pig, and this just shows inflammation after a myocardial infarct. We can label that with perfluorocarbon and image that, and so we're confident about the technical aspects. Our delivery is going to be using magnetic cath tracking, where we inject the cells directly into the myocardium. And so this is the infarct here. These are the injection sites. This is a prior study that we did using MRI. What we like to be able to do now with this is the next phase, is basically follow the fate of these injected cells and count how many are there over time. So my lab now is focused on improving the detection limits using this. We'd like to make this perfluorocarbon cell tracking technology the 2.0 version, essentially, bring it to the next level of sensitivity. The way we're doing that is we're working on the process. We've been working with GE Healthcare to develop cell labeling at bulk at a billion cell scale using these bioprocessors. And so this is ongoing work. We're also developing pulse sequences that can greatly accelerate the acquisition of the fluorine-19 signal. So this example, 54 minutes versus 7 minutes, essentially the same signal-to-noise ratio. So all we did here was just implement physics, and this allows us to image the cells much quicker. We're also working on developing new molecules, and this is a very exciting area, working with Roger Chen's lab to make perfluorocarbon molecules that have enhanced sensitivity. And this is work of Alex Kislukin, who's working in our lab. And he's been engineering perfluorocarbon emulsions with metals incorporated into it that greatly accelerates the T1 relaxation rate by orders of magnitude. And you can get a great enhancement of sensitivity over the agents that we're currently use that look like this. And these are the new agents here. So they're much brighter. You can image fewer cells. There's also possibilities for hardware optimization. Overall, we believe that you can achieve 100 times reduction in imaging time. And this implies about over a 10-fold improvement in cell detection. And so these are some of the activities that we're working on in our lab. So you can see it's very integrated. It involves principles of chemistry, biology, physics. We try to integrate all of that toward this goal. Thank you very much.
Thank you. I have two quick questions. One is, what is the half-life of the compounds? And the other is, what happens at cell death? Are they taken up by the surrounding tissue?
Yeah, these materials are indelible, so they label only viable cells in culture. Cells that are apoptotic are not labeled. Once they're labeled, as long as the cell remains viable, the cell will remain labeled. The cell does not degrade the material, nor does low pH degrade it, nor does enzymes inside the cell degrade it. So it's long-lived. If the cell dies, the material is released. The clearance through the body of those emulsion droplets are through the RES. It's taken up by the RES, and it's ultimately cleared through the body through lung exhalation. That's the pathway. It's a little unconventional.
So in terms of the labeling, with autologous therapies, there's issues of the engraftment proliferation and differentiation. So when the cell is proliferating and also differentiating, does it maintain the stability of the labeling, and are you able to track that still?
Yeah, I mean, if the cells proliferate, there'll be a dilution. Half the dose will go to the daughter cells, thereby diluting the material. If those cells stay in the same place, you'll still see the same amount of material, but you'll get a diminishing of the accuracy of the cell quantification. Okay? Differentiation, we can't say anything about. It's not a smart agent in that sense. So.
Quick. So, use of the VeriCell product after it's injected into a patient's heart, is it possible to track the secreted factors, like the extracellular vesicles that are excreted from the cells?
That would be great. We don't know how to do that right now, but that would be great. But it's not possible with this technology.
Great. Thank you very, very much, Eric.